Okay, Professor Esserman, nice to meet you online.
Nice to meet you too.
First, congratulations for your publications on JAMA Oncology. It's an impressive trial, I-SPY 2. We know that the I-SPY 2, you in the publications, it shows that the pCR rate is very a known prognostic biomarker for long-term outcomes. And I-SPY 2 also evaluated if the strength of these clinical association persistent in the context of this phase 2 new adjuvant platform trial. And we also find that this study enrolled some high-risk breast cancers, and we also find that the participants were adaptively randomized to one of the several different investigational regimens or control therapy within molecular subtypes for almost 6 years. Also, the analysis included participants with follow-up data available as of 2019. So you are the PI of this trial. Could you introduce us some key points or key elements of this trial, please?
Certainly. So I-SPY 2 is really a trial for women with high-risk breast cancer. So all the patients had molecularly high-risk tumors. These are people who have the highest risk to die of their disease within 3 to 5 years. That's actually really important because that is these are highly proliferative tumors. And I think there's two things that are really particularly striking about where we chose to do this work. The first is that we really wanted to do this work in patients who were early stage but high risk. And the reason for that is because when we test agents in the metastatic setting, now we can often prolong survival, or we can at least prolong, delay progression, and maybe sometimes we can prolong survival for some period of time, but for the most part, we're not able to cure people in the metastatic setting. So one of the things that we were very interested in is moving drug development to the earlier stage of disease. When there are promising agents, why not give it to people who have the highest risk for dying and intervene at a time when we can actually save their lives? So the important, I think one of the most important things about I-SPY is that the whole goal was to demonstrate that we would be able to improve event-free survival and distant recurrence-free survival. The trial is powered around an early endpoint, which is complete response or disappearance of the tumor at the time of surgery. And the reason for that is because we had actually, in I-SPY 1, we had really demonstrated this is likely to be the most important subtype, and that it mattered, but that the most important endpoint, and that it matters by subtype, that each subtype has a different rate of response to therapies. And we thought that this would be a platform for really trying to make a difference in early stage breast cancer. So one of the hallmarks of the trial is that it is a platform trial. The trial is the basic principle of putting drugs in combination with Taxol or some combination of drugs prior to giving AC, using an early endpoint of pCR. And what happens is, for the drugs that are in the trial at the time, if one drug appears to be doing better than the others, the next person who comes in with that subtype is more likely to get that agent. So that's important, adaptively randomized. Another very important aspect is that it's a biomarker-rich trial. And by that, we mean that we are trying to understand how to better classify tumors, to really understand more about predicting who's going to respond. So we had a lot of qualifying biomarkers, ways in which we thought we could classify the tumors that would make a difference and improve our chances of understanding who would respond to what therapy. And the last thing that's really important is this is guided by imaging. So we do frequent MRIs before surgery, before starting chemotherapy, at 3 weeks, at 12 weeks, and 24 weeks. And the idea is that the MRI and we use a functional tumor volume developed by my colleague Nola Hilton. And the whole purpose of that then is to be able to have yet an earlier endpoint than complete response, to look at volume response so that the trial can learn as we go and keep assigning agents. So we're about to put, the trial is still ongoing. We're actually about to put our 24th combination into the trial. And this landmark study demonstrating indeed that regardless of subtype, regardless of novel agent, pCR is incredibly prognostic. What this tells us that our future trials should be organized so that we drive every patient to getting a pCR. So that's kind of where we're headed next as we evolve and learn from this trial.
Great, that's an impressive results. And we also see this trial as a landmark study. We also see, I still have some questions maybe. The first is that for this trial, published on JAMA Oncology, only showed that the pCR is a well-known prognostic biomarker for long-term outcomes, just like the CT NeoBC pooled analysis showed that. Actually, we found that some pCR rate is not as good as, or not more higher than previous studies, such like in Jian study, the pCR for the ER negative and HER2 positive patients, we also we can get 89% of 89% pCR rate, and also for the ER positive and HER2 positive in Jian study, we also get 55%. But in this clinical trial, we only find for these two populations, 68% and 40% pCR rate. Do you think this adaptive trial and find any newer or newest drug for the combination of previously treatment to increase the pCR and increase the EFS? Do you think you have got a good result for that?
Well, you know, it's interesting. I think that people have said if you look at the controls, part of this is I think that we certainly are, again, it depends who your patients are. The 89% rate, I'd be interested what you're using to get that. To get in the trial, you have to have a tumor that's 2.5 centimeters or larger. So these are 2/3 are node positive to start, at least. And we actually have some new data looking at circulating tumor DNA, and 75% have circulating tumor DNA as well. So it's a pretty high-risk group of patients. The highest response rate that we have seen with any new agent is 60% with pembrolizumab combined with Taxol. That's the highest we've seen so far. And we, in the triple negative rate, and the control is really, closer to 29 or 28%. And it's been pretty consistently that. That does not include carbo. We know with carboplatin, the veliparib carbo that graduated is one of the first combinations. In a subsequent study was shown the same 50% response rate was obtained, but it was really largely, essentially all due to carboplatin. So I'd be very interested to know what you're using to get an 89% complete response rate. And then I'll make one more comment about that. What are you using to get that?
That means if we use the carboplatin for the I just mentioned that the triple negative breast cancer in the CIGB 1493, we got a PCR rate about 60%. So for the triple
They were
negative,
about 50% in the NCLGB. So what? So but you said that you are observing 89%.
oh, that means ER negative and HER2 positive for
Triple negative.
Trento study. For Trento study, HER2 positive, ER negative. The PCR rate
Oh,
is
so in the HER2 positive hormone negative rates with THP, and that subset were in the 80 to 90% rate as well. That's another one that graduated. So it's quite high. Not as high, more like 60, 65% with the hormone positive, HER2 positive. Another thing that we've done that I think is really interesting in the trial is we've really standardized the way in which we evaluate all the outcomes. So all of the pathologists use the residual cancer burden method. There's two reasons for that. In I-SPY1, when we didn't have a standardized way of evaluating the results, when we went back and had all of the cases evaluated with the RCB method, we dropped our complete response rate from 35% to 27%. So I think it matters how you do it. So there are several. So the surgeries, even across our 20 sites, we've really worked on standardizing the way people do things, standardizing the way the MRs are done, and standardizing the way the pathologists read them in. So bread loafing all the specimens, really measuring the tumor bed, and then looking at both cellularity and looking for the clips. So if someone has a complete response, we also don't do mastectomy very often in that setting. And we go after the original tumor site by locating the clip or the original disease, and then making sure that we really have targeted it. So I think that and, using, we've actually gotten away from doing complete axillary dissections, but at least looking to do the removal of the original positive node, the clip node. So we have to demonstrate that you can do that. I think those are things that help you keeps your data as honest as possible.
That's great, I think. So, Professor Esserman, do you find any new drug to get a higher response, a higher PCR you have get, and maybe you can use a seamless design for these drugs to a phase 3 study? And do you find any drug to show us for that?
Well, so we are in the process of trying to transition the trial. We have a big grant or big study to try and transition to something that allows us to individualize patient outcomes. And what we would like to do, hopefully by next March, the trial will transition to starting with non-chemotherapy combinations, looking to see how many people can get to a complete response, and we can evaluate that with a combination of a biopsy and MR volume change. And that's actually in that tool to assess interim response and allow patients to go to the OR if it looks like they've gotten a complete response is in place. So we're testing that now. And then the idea would be, if you don't get a complete response, you are rescued by some combination with Taxol that would be the best that we've seen in I-SPY to date. And then if you still have, then again, if you have a complete response, you can go to the OR, and only then would you wind up with AC. And that way, we hope that we will be able to reduce the number of patients who are exposed to drugs like adriamycin. It's a very good and active drug, but it's actually fairly toxic. So I think people feel better about using it if it really is needed. And I think over time, we're going to find things that will be better than AC. And I'm looking forward to being able to randomize when that's possible. The HER2 subtype is probably the subtype that's the closest to being ready to have a series of agents that you can use that would be where people would get to complete response 80–90% of the time without, and still have maybe some drugs that you could use that would be preferential to AC. So that's the new idea. Imaging will play an important role in that. We believe ctDNA will, like circulating DNA, will probably play a role in that. Increasingly, we're seeing non-chemotherapy combinations with antibody conjugated toxins, with their combinations that we think can, potentiate the immune response. There are combinations that can, lead to increased DNA damage repair that could be combined with things like PARP inhibitors. So we want to encourage the industry to be creative and thoughtful and to continue to develop their best drugs. We don't have to, I would imagine that pembrolizumab or one of the PD-1 inhibitors is going to wind up getting approval based on the keynote. Merck took the data we had in I-SPY 2 and designed a keynote 522, but they added carbo. So it's Taxol, carbo, pembro, followed by AC pembro, followed by surgery and pembro, or pembro. I don't know that everybody needs that. I think that pembro 4 actually works pretty well too. But on the basis of both the I-SPY data, then followed by their confirmatory trial, I would imagine that drug will get approval. I see there's a PDUFA filing for March of next year. And so then I think you have to say, well, is that it? Is that the end of all the innovation? And I would submit that no. First of all, 100% of people aren't getting a complete response. And second, everyone's getting everything. That's not the future of personalized medicine. We want to find out who needs what. There are some really great responders. Let those people have less therapy. And those who are intermediate can have some more, and those who really need the most get, all 3 cycles. But I think that we're excited about that idea.
That's great. I hope more and more new drugs are designed because of your trial, for the industry to design new drugs for our patients, I think.
I agree. And if you don't have trials that allow them to put innovative drugs forward, and we don't allow people to test them, then we're not going to, then we can't have that future.
Yes. So I think the platform is very great because you can enroll the new drugs. We need no any new clinical trials. I think this is very good. So actually, this is an umbrella study. You focus in all the patients' breast cancer, breast cancers. You just mentioned future. Actually, our cancer center from Fudan University from Shanghai, a study named just a future, designed by our Professor Shaw, had published in Cell Research. We focused on the metastatic refractory triple-negative breast cancer. We designed 7 arms, and we also find that this phase 1b/2 subtyping based and a genomic biomarker guided umbrella trial, we want to evaluate the efficacy of these targets. We designed the Fudan for subtyping for the triple-negative cancer, and we use this for subtyping design this clinical trial. We found that immunotherapy in particular achieved highest over response, almost more than 60%. And we also find the HER2 mutated patients. We use the HER2 TKIs and got 100% of response for triple-negative cancer. And I think maybe actually, Professor Esserman, in the future we can design some focusing the triple negative cancer for drug testing. I think
Definitely.
maybe in the future, do you have any design for the I-SPY 3, I-SPY 4? Any
Well, I call it I Spy 2.2.
2.2?
So that we can continue to use all the data that we've accumulated. And we, again, especially if you're thinking about trying to optimize people's outcomes, who wants, the idea is you should try and get the best for each person. So it's hard to have it be a control, but if we use the whole database that we've had, we can continue to use those, I would say, they're not that way. They're not historic controls. They're actually contemporary controls, with similar agents. So that as we change, that we can really compare. I think the other place where I think we need a lot of improvement is in the endocrine positive group. So that we still, I think that there are, we just presented at AACR the durvalumab plus olaparib arm with Taxol. And what you see is that there's a subset of the hormone positive patients that are more basal-like, and those respond nicely to the IO drugs. But the ones that aren't still have a pretty dismal pCR rate. I mean, if they get there, they do well, but it's not great, and we have to do better there. And so we are looking for different kinds of combinations and trying to understand what is making those patients resistant, as they really don't do well. Just like if you have a lot of residual disease left in your hormone receptor positive, it's not good. I mean, people do very badly at 3 years, so we have to do better. And then as well, all the patients that screen out that are molecularly low risk, but clinically high risk, we need to find better treatments for them as well. So we're starting an endocrine optimization pilot. I'm thinking about SERDs as backbones and trying to look at combinations of agents that might do better in that setting. And we may allow some of the borderline cases for molecularly high risk to be eligible for that as well, so that we can try and understand how to improve outcomes, or if there is. I think the hard part is we don't have a good early endpoint. So that's part of what the first phase is going to be about, trying to come up with an endpoint that we think there's a good dynamic range could really help us advance the field that way.
That's great, I think. And maybe the last question. You published papers that showed that hazard ratios for PCR versus non-PCR were 0.19 for EFS and 0.21 for DRFS. And I just wonder, for non-PCR patients, do all these patients receive actually such like the Catherine study for the creative study treatment, and if they have do this treatment, I think this hazard ratio is very impressive. Could you give some data for us?
Yes. So when the study started, the chance that someone with non-PCR would get therapy was less than 7%. By the last year of the study, so this is the time that we finished this, so this is the end of 2016, when this last group is 25% of people were getting adjuvant therapy. And, it's interesting, the statisticians will often say, well, you haven't proven surrogacy because you didn't show in every arm. And, in I-SPY, each one of the arms isn't that big. So you can have variation. And so we may not have proved surrogacy, but I can say that clinically in the United States, most people will give additional therapy when patients don't achieve complete response. That's a standard in HER2. It's a standard in triple negative disease. and I think is actually becoming standard in hormone positive patients. So the clinicians have voted with their, we have a saying, they vote with their feet, meaning if you watch what people do, that's whether or not people believe the data. And they do. And there are now many post-neoadjuvant studies showing that you can do better. Actually, the Penelope trial will be out shortly as well. And that was, I think, randomizing people to palbociclib or not. If they're hormone receptor positive, we have the study of TDM1 that showed efficacy. And there's Xeloda. I mean, so there's many options for patients, and most patients are getting it. So I think that also says that in the future, and we'll see, we're just about to do a follow-up on all our patients up until through the current time, and we're going to write a paper on looking at the trends in the use of adjuvant therapy. I think it's, you know, it was our hope to change practice and get people to use neoadjuvant therapy, which I think we're much closer to that now than we were when we started. Very few people were getting it in 2010. I think most people with high-risk disease are getting it now. And the fact that we've shown that it really works, even across novel agents, I think then people, I think, rightly say, wow, that I should do better for my patients and try and get them to a better outcome. So I think that's one of the reasons why we feel like we're going to need to make some adjustments to the trial. We'll call it I-SPY 2.2, and try and give people 3 shots on goal, 3 different regimens. But we'll allow people who have a complete response, who are great responders, to stop. There's a big meta-analysis that Laura Springer et al. Did with Didier Bardia with 27,000 patients. And again, it shows the same thing, that PCR is a great predictor of outcome. They didn't have a lot of novel therapies, but the other thing they showed was that once you got a PCR, there were 8,000 patients who got the first regimen, then were randomized to more therapy versus not after surgery. And it shows that once you have a PCR, your outcome isn't improved by adding more therapy. So I think that's also really very important. And I think that makes us say, well, once you've gotten to a PCR, that we can stop. And I think that will also allow us not only to improve efficacy, but decrease toxicity.
Okay, this is the last question. Okay, thank you for for this interview, Dr. Esserman. According to what you said, I think this is very helpful for all audience to understand what you published in the JAMA Oncology. This great paper, and we also eager for the results of you have designed the I-SPY 2.2, and we also eager for maybe some results for the PCR more than 90% for all patients. I think maybe we can meet next year, and we found what
hopefully in person, that would be good. I would say one thing. The other thing that we know is that that the amount of disease you have left is also very important. We have a paper under review right now that shows that the residual cancer burden adds a lot of value. So it's not just PCR, but in fact, if you have a near complete response, you do much better than if you have a moderate amount of disease. And if you have a lot of disease, you do really very badly. So it's not only important to get people to PCR, it's important to get people off the RCB3, out of the having a lot of disease left. And one of the great things about I-SPY is that we have now about 150 of these patients where we have serial biopsies and the serial ability to really do a deep dive on the biology of these resistant patients. So we hope that will give us some ideas. And if your trial in Shanghai has some good ideas and you think there's some things that we should be trying in the early setting, please email me. We'd be really so interested in your results to help us design better regimens and get them to patients earlier.
Okay, great. Oh, thank you, Esserman.